Okay, today we're going to review transcription and translation. If you'll open up your EOC review packet to page 10, at the end of today's lesson, I want you to submit pages 10 and 11. So take pictures of those two pages and submit that into Canvas. And then I would also like for you to take your EOC review quiz number 5. Please do that before you leave today. Okay, so when we look at our DNA chart, you're given one side of the DNA, ACG. Okay, again, remember your base pairing rule, adenine bonds with thymine, cytosine bonds with guanine. So the other side of the DNA would be TGC. Now, you have to remember that there's no thymine in RNA. So whenever you want to write a thymine, no, uracil is going to take its place. So again, when mRNA gets the code from DNA, that is transcription. So what bonds with T? A. What bonds with G? C. What bonds with G? C. Okay, so when we look up ACG in the chart, okay, so the first base is A, that brings us to this row. The second base is C, that brings us to this box. And the third base is G. So ACG codes for threonine. Now I'm going to stop the video, I'm going to pause it, and I want you to do the rest of the columns. Okay, welcome back. So to check your work, if one side of the DNA is AAG, the other side would be TTC. Again, finding the mRNA would be AAG, and if you look up AAG, that brings us to lysine, the amino acid lysine. The next row, if one side of the DNA is TGG, the other side of the DNA would be ACC. Then the mRNA would be UGG. Now, if you put a T there, you need to make that correction. There is no thymine in RNA, uracil takes its place. And then if we look up UGG, that brings us here, which is tryptophan, TRP. The next column, if one side of the DNA is CCT, the other side would be GGA. The mRNA would be CCU, again, U. So if we look up CCU, it codes for proline, the amino acid proline. And then lastly, if one side of the DNA is GAA, the other side of the DNA would be CTT. The mRNA would be GAA. And if we look up GAA, it codes for the amino acid glutamine. So it's very much fair game on the EOC for you have to be able to read this chart. Remember, this is a chart of codons. So you have to look up the mRNA codon to find the amino acid. You can't look up the DNA. It has to be converted first to mRNA and then look up the amino acid. Okay, so now I've got a little picture drawn, and again, you can pause the video to take a moment to draw this. So here I've got mRNA, and I've got several codons, AUG, GGC, UUU. And remember, mRNA got the code from DNA, okay, that was transcription. So now we're going to be at the ribosome where translation takes place. And up here I've got transfer RNAs. And remember, transfer RNA has got these triplets called anticodons. So UAC, CCG are anticodons. And the job of transfer RNA is to deliver the amino acids to the ribosome. So which anticodon is going to come here and base pair with AUG? Okay, this one would. So UAC, that anticodon, will base pair with the codon AUG. Okay, the next, what anticodon will base pair with GGC? CCG. So it will then come in. Now, when two amino acids get side by side, a peptide bond will form between them. So this red line is representing a peptide bond. This will continue until a stop codon is reached. And remember, there are 3 stop codons, UAA, UAG, and UGA. And then the protein will be released and we're finished. So here I've kind of color-coded some things in our picture here. So the DNA is in blue, the mRNA is in pink, and the tRNA is in green. Okay, so here, when mRNA is being made from DNA, that is transcription. And remember, that happens in the nucleus of the cell. Once mRNA gets the sequence from DNA, it's going to go out here to the ribosome. So this is going to be on a ribosome out in the cytoplasm. So the pink is my mRNA. And again, these three triplets are codons. Here's my transfer RNAs. And these are amino acids. Okay, so all these circles are amino acids. When the two amino acids get side by side after the anticodon base pairs with a codon, remember a peptide bond will form between them. Okay, so again, this is transcription, the making of mRNA from DNA. The making of the protein from mRNA is translation, So to describe that, we've got a little paragraph, and I understand that it's long, and you can stop and start the video as you need to. But question 8 is asking, describe what is happening in the picture above. Okay, so during transcription, mRNA gets the code, remember that's the order of nitrogen bases from DNA in the nucleus. The mRNA moves out of the nucleus to the ribosome. Then anticodons on tRNA will carry amino acids to the ribosome. Anticodons will base pair with codons on mRNA. Once the two amino acids are beside each other, a peptide bond will form. This process of combining amino acids together to form a protein is called translation. That is called translation. So again, take a moment. Again, feel free to stop the video so you can make sure you get everything down in the paragraph. Okay, lastly, I'd like to spend a moment talking about this last little bit. So again, here's another picture, just another small picture of translation. Again, here's your mRNA. Here's the tRNA. So again, we've got the anticodon ACG. And then the anticodon UGC is going to base pair with it. Here's your transfer RNA, and it's carrying amino acids. Again, when two amino acids get side by side, a peptide bond will form between them, forming the protein. So in question number 9, it says, refer to the diagram above. What type of organic molecule is being synthesized? Okay, well, I'm joining amino acids. What organic molecule is made up of amino acids? Protein. So protein is the type of organic molecule. Remember, proteins are made up of amino acids. Again, refer to the diagram above. What process is being shown? This is the process of translation. The making of a protein from mRNA is translation. Okay, so again, now if you will take pictures of page 10 and page 11 and submit those pictures, and then take the quiz number 5. I hope y'all have a great weekend. Bye.